Tom Tuschl was a postdoc, and he had been for about a year and a half, shared between David Bartel and myself. And Phil Zamore was a postdoc with David after a number of changes. And Tom and Phil were interacting because they were both RNA biologists as postdocs and very good people. And I had been talking with this perspective. I wrote the perspective to motivate the issue of can we use RNAi to perturb genes in mammalian cells. So we published the paper December 1999. It's the first paper we published. And we made it very specific. We got it there by December 99, so it would have a 99 reference.
And which publication is that?
That's Genes in Development.
Okay.
And Tom had finished the project he was basically working on. He had a job in Germany. He had about 3 months left here. And he came into my office and we were talking about finishing the paper. It was basically gone. He had the job. So he had 3 months. What was he going to do? And he basically said I could, we were talking, I could finish up some details on this paper and then go off. And, I was big on this RNAi thing. And I have, in my life, I've made a real living off developing the biochemistry of these complex processes. So I said, why don't you try to develop the biochemistry of this RNAi in vitro and do it in an extract and test it and see if we can see it. So he went off and collaborated with David, talked about it, and Phil Zamore, but more importantly, Zamore. And Zamore had already made extracts in frog oocytes. These are the eggs of frogs, and they're very big egg cells, and you can make an extract out of them, and they got all sorts of goodies in them. And we knew already from the literature that if you injected RNA into them, you could see some RNAi. And the first experiment, it was designed beautifully, the first experiment worked. And he came in on a Friday afternoon, just as I was getting ready to leave, and he said, I think it works. And he showed me the design of the experiment and the signal and the controls, which shouldn't work, didn't work, and the one that did work, did work. And he said, we'll now begin to play with this extract. And came back the next week, and there was a big whopping signal. So we knew we had the ability to go from double-strand RNA to silencing a messenger RNA in a test tube. And we worked, that was in Drosophila. So we worked on the Drosophila system to the point where we knew that it generated small RNAs. The bad news was Tom had to go off to Germany, to Max Planck, and Phil had to go to UMass. So now we had Tom in Germany, Phil in UMass. We knew we had one of the more interesting breakthroughs in science biochemically in our midst. And Bartel was running a big lab. These two people had all the expertise. So Tom went off to Germany. And it was in Germany that he cloned these small RNAs and created then the first siRNA type structure. And Zamore collaborated with him out of UMass. So then Tuschl in Germany in 2000 and 2001 elucidated by cloning the siRNA structure, and Phil Zamore extended and collaborated with him. And that's why there was the UMass part of the patent and the Max Planck part of the patent and MIT part of the patent. So we had all of these patent pieces of this patent. Each of them had patents, and MIT then got them to agree that MIT would negotiate the patent with each getting their respective rights. Then when Tuschl did that cloning and we had the siRNA structure, he made siRNAs and showed they were active. So we got together and decided, we talked about this. It certainly was a very important technology for studies in laboratories. And we wanted to patent it so we could license that patent to get laboratory supplies. We had to patent because Germany wanted it in Europe. So we had to license somebody in Europe. We licensed someone in US. So we decided we'd license about 3 people. And that we had a license, and we licensed it to MIT, each of them getting their royalties. And then in the clinical side, we reserved the rights. And we then started talking among ourselves about how to translate this. And I had been in Biogen, and this is the most exciting piece of science that I thought had translational present that I knew of. So we all agreed we would participate in starting a company to translate this. And we invited Paul Schimmel in because Paul knew a lot more about negotiating with venture, because he'd been involved a long time. So I wanted to know how the pharmaceutical world, I wanted to do 2 things. I wanted to get a reaction from the pharmaceutical world, or biotech, and 2, I wanted people to know we were thinking about this. So I contacted people in Millennium, where John was at, and I may have even gone to John to make that contact. I asked if we could come as a group and make a presentation about RNAi. So we went up there and made a presentation. We were working with Lida Nelson at MIT. It's somebody we should celebrate. Well, she's no longer associated with MIT. She retired, but she's still active in the community. So we had talked about it. We actually had talked to one venture capitalist that was introduced to Lida. I contacted Paul. Paul asked him about venture capital, who he thought was good to work with. And we got Polaris, and we ultimately got Cardinal. But we went up to Millennium and made a pitch. And then Millennium said, which is what I expected at the stage they were just becoming commercial. We can't deal with this. Right. Is too long term. It's too much. It's a diversion. But then Rachel called shortly thereafter. Once we then picked Polaris to be our, and Christoph Westphal at that stage was the venture partner at Polaris. And John Clark, the head of Cardinal out of Princeton, were the 2 that we selected to be our venture partners. Then John was contacted and became the CEO about 3 months into the organization. Rachel Myers was hired at the same. She was a grad student with me. A lot of the Biogen people, because John was at Biogen before he went to Millennium and then reappears here. So